You know, every living thing, from a single blade of grass all the way up to you and me, is built by this incredible process called cell division. But here's the thing. It's not a one-size-fits-all deal. It's really a tale of two destinies, two totally different paths a cell can take. So how does a cell choose whether its job is to build a body or to create a whole new life?
I mean, really think about that for a second. That one cell has this fundamental choice to make. Is it going to make a perfect, exact copy of itself to help us grow, to repair damage, and just maintain the body? Or is it going to shuffle its genetic deck to create something brand new for the next generation? That's the big question we're diving into today.
All right, so here's our game plan. We'll start with those two destinies. Then we'll get into the process for making perfect copies, which is called mitosis. After that, we're going to unpack the much more complex process for creating variety, meiosis, and we'll split that into its two major phases to make it super clear. And finally, we'll wrap it all up by looking at why this stuff is just absolutely crucial for life as we know it.
Okay, let's get right to the heart of it. This fundamental choice that is really the foundation of all of biology.
Honestly, the best way to wrap your head around this is with two really simple analogies. On one side, you've got mitosis, which I like to call the clone engine. Its job is to just crank out perfect, identical copies with ruthless efficiency. But on the other side, you've got meiosis, the variety generator. And this one is all about getting creative, mixing things up to produce unique genetic combinations. Two very different goals, and so two very different processes.
So, let's start with that clone engine. This is the process that literally builds you and keeps you running every single day with just incredible precision.
The official term for this is mitosis, and the definition is actually beautifully simple. It's what we call an equational division, which is just a fancy way of saying what you start with is exactly what you end up with. One parent cell, with its full set of genetic instructions, divides into two daughter cells that are perfect, identical copies. No changes, no surprises. So why is this so important? Well, for starters, this is how we grow from just a single fertilized egg into a complex person. But it's also absolutely vital for repair. Think about it. The cells lining your gut, the outer layer of your skin, your red blood cells, they're all constantly wearing out and being replaced. That's mitosis at work. It's like the ultimate maintenance program for your body.
So if there's one thing to remember about mitosis, it's this number, 2. The end result of one round is always 2. Two completely identical daughter cells, each with the full set of chromosomes, all ready to do their job in the body.
Okay, now let's completely switch gears and talk about that second destiny. Because if mitosis is all about preserving the present, meiosis is all about designing the future. This isn't about maintaining one body, it's about creating the blueprint for a whole new one.
And that process is meiosis. Now, unlike mitosis, this is a reduction division. Its entire purpose is to take a special diploid cell, that's one with two full sets of chromosomes, and cut that chromosome number in half. The result is what we call haploid cells, with just one set of chromosomes. You probably know them better as gametes, or sex cells, like sperm and eggs. Now this is where it gets really interesting, because meiosis is different from the very start. First off, it involves two separate divisions, one right after the other. But, and this is so important, the DNA only copies itself once, right at the very beginning. And maybe most importantly, it involves similar chromosomes pairing up and doing something called recombination. Trust me, this is where the magic happens.
So let's zoom in on that first division, Meiosis I. This really is the main event. I like to call it the Great Shuffle, because this is where pretty much all of that amazing genetic diversity comes from.
The first part of this, prophase I, is super complex, so scientists break it into 5 stages. Now don't worry about memorizing these names. What's important is the story. First, the chromosomes pop into view. Then, they find their matching partner, the one from the other parent. And then, in this really critical packaging stage, they literally swap pieces of genetic information. That's called crossing over. The last couple of stages are just about getting the cell ready to divide. But that swap, that's the whole point. So let's just focus on that one key event for a second, crossing over. This is where chromosomes from your mom and dad physically get together and swap sections of their DNA. A piece of one chromosome literally breaks off and attaches to its partner, and vice versa. This is the engine of genetic variation. It's a physical reshuffling of the genetic code. It's incredible. So after all that complex dancing and shuffling in prophase I, the goal of this whole first division is actually really simple. Just separate those homologous pairs.
One chromosome from each pair gets pulled to one side of the cell, and the other one goes to the opposite side. That's it.
All right, now we're ready to enter the second phase, Meiosis II. And hey, if you understood mitosis, this next part is going to look very, very familiar.
Yeah, Meiosis II is pretty much a mitosis division. The two cells that we made in Meiosis I now divide again. But this time, the goal is different. We're not trying to separate homologous pairs. We already did that. The goal now is to separate the sister chromatids, those two identical copies that make up each individual chromosome.
And what's the final payoff for this entire two-step process? 4. You end up with 4 unique daughter cells. Each one has only half the number of chromosomes as the original parent cell. And get this, none of them are identical to each other, and none of them are identical to the parent cell that started it all. This is the raw material for the next generation.
So there we have it, the clone engine and the variety generator. Let's bring this all home and talk about why these two processes are the absolute foundation of all complex life on this planet.
This chart just lays it all out perfectly. You've got mitosis for growth and repair versus meiosis for sexual reproduction. One division versus two, an outcome of 2 identical cells versus 4 unique cells. But the most important difference, the real game changer, is that last row. Mitosis creates 0 genetic variation. Meiosis, on the other hand, creates a massive amount of it.
And that right there brings us to the ultimate point of all this. That variation created by meiosis isn't just about making unique individuals for fun. As our source material puts it so well, variations are very important for the process of evolution. Without this constant shuffling of genes, life just couldn't adapt, it couldn't change, it couldn't evolve.
So I'll leave you with this final thought. Mitosis is the process that builds and maintains our bodies in the here and now. But meiosis? Meiosis is the process that's constantly designing the future of our species. It's always experimenting, always creating new combinations. Which just makes you wonder, what new variations are being shuffled right now, and where will evolution take them next?